Okay, well, welcome everybody, and it's my pleasure today to introduce Christian Gut from the University of Siegen, who will be giving today's colloquium. And in the area of, he's been working in the area of condensed matter physics and statistical mechanics, experimental work, and he has been doing a lot of work both in the hard condensed matter physics related, for example, to spins or ferroelectrics, as well as in the area of soft condensed matter physics, and that's what we're going to be hearing today. A lot, as you can see here from the title, related to crowding, hydrodynamic effects, and protein motion. Before we start, I just want to say a few words about Christian's background. So he did his bachelor, master, and PhD in experimental physics in the University of Kiel, and then postdoc work in Dortmund and University of California in San Diego. And then back to Dortmund for his habilitation, and he also been working as a senior scientist in DAISY. And since 2013, he's been professor in experimental physics in Siegen. He also holds several important positions in the community. He's the chair of the Committee Synchrotron Radiation Research in Germany. And also he's in a scientific advisory board at MAX4, at ESRF, and Petra 3. And that is some of the few positions that he holds. So he's here now for the whole week. So if somebody wants to arrange and meet with him, please reach out to me, and I can arrange that. We can also, if you want to show him your lab and so on, please contact me and I'll be happy to arrange that. So without further ado, I pass the word to Christian.
Yeah.
Thank you.
Yeah, thank you very much, Vivas, for having me here and for giving the opportunity to talk once again about science, which I really appreciate, I have to say, because we were talking so much about large-scale facilities and high-tech agenda and funding things and data over the last year. So it is good to concentrate back on science. So I want to show you a little bit the results which we were getting over the last 4 or 5 years together with Vivas groups here in Stockholm, making use of the unique capabilities of a large-scale facility, which is the European XFEL in Schenefeld in Hamburg. And the topic which I want to address here is slowdown and diffusion of proteins. So these are typical group which is involved in these experiments. You can see those are larger groups, larger experiments. So we organize that with 30, 40 people typically. And this here is a sketch of what we are interested in. So we are a university group. So we have to see how we can organize funding and how we can also organize samples which we can afford. And in the first experiments, we were using extremely radiation sensitive samples from a collaborator in Sweden, which will not be named, but those samples have been burned very, very quickly under the beam. And so we were thinking about, well, what other proteins can we use? And egg is something which the university group in Germany can easily afford. And so we are going to the gas station and get these eggs here. So that is a very good and very nice sample system to start these experiments with. Those are the groups which are involved. So we are running something which we call a long-term proposal. That means every 6 months we have a beam time at European XFEL. And as you can see here, that's the University of Siegen. This is my group. Then we are together with Tübingen, with Vivas group here in Stockholm, European XFEL, Dortmund, DAISY, ESRF is involved. So it's a larger collaboration here. And most importantly, how that started is something, a funding scheme which we have between Germany and Sweden, which is called the Röntgen-Angström cluster, which allowed us here together with Vivas to start all of this. All right, so that is really a success story, and we hope that in some sort we can continue with these kinds of German-Swedish collaboration. I think that would be very, very important. Okay, so the outline of the talk, I would like to introduce a little bit the scientific topic, diffusion and crowding. Why is that interesting? Why do we want to follow that? And then, of course, as we are large-scale facility aficionados, I would like to tell you a little bit about how we do these experiments at the European XFEL. And the idea of these large-scale facilities is, of course, is to invent new methods and make use of these very expensive machines. And then I have a couple of examples. We have to see how many of those I can really cover. but prominently aggioic, LDL dynamics, ferritin, which is another kind of protein in self-crowding, and then ferritin in polymeric crowders. And I would like to close then with an outline and perspective. So the scientific paradigm which we are interested in is the so-called crowding in biological environments. So if you think about the cell and the proteins inside the cell, then these proteins are not in a diluted condition, but this is a very highly crowded environment. So these volume fractions of proteins here can reach 20, 30, 40%, and that is a computer simulation about proteins moving in such a highly dense environment, right? And then you can see they are performing Brownian motion, but they are not performing Brownian motion like in a textbook. And that is because there's a next neighbor around where they are, so to say, bumping in. And this very crowded environment is, of course, having effects, so to say, on the biological function. So this regulates a lot of physiological properties in the cell, like, for example, diffusion and activities, enzymatic activities, signal transmission, phase transition. So it's really important to understand that this is not a diluted environment here, but this is a highly concentrated environment of proteins. And obviously, this highly crowded environment is, so to say, steering and affecting somehow most of the biological reactions which is happening. So there's since 20, 30 years, this paradigm of crowding is very well known, and people are interested in understanding what are, so to say, the consequences of this crowding. And one of those consequences which have been early understood is that if you look into the diffusion of proteins in cells, then what has been measured many times is a reduction of that diffusion, that means of the velocity of these proteins, by a factor of 10 or even more compared to crowded solutions. And the big question which we try to answer is, what is causing this reduction of a factor of 10, right? And there was a beautiful chemical review paper here in 2024, just 2 years ago, from Annalisa Pastore here, who is, so to say, who was looking into the details and going through the literature for the last 20 years. And what is basically, so to say, the overview of that is that viscosity, volume exclusion, and molecular interactions are the driving factors for reducing diffusion, right? Excluded volume is clear that when the volume is getting more and more rare in the cell, then of course the diffusion is reduced, that is clear. But the question is, what are other interactions doing in that respect, so to say, right? And that is, as I was saying already, that is has consequences for protein folding, for stability, for aggregation. So it is important to understand why is that happening here, right? And of course, that is a biological topic. So the biological groups are very much interesting in that, and they are very much doing lots of measurement. We are physicists, so we try to understand it in a quantitative behavior, right? We really want to understand what is quantitatively happening here in terms of physics, what is reducing the diffusion, what is reducing the velocity of these proteins. And one of the things which we are looking in detail, I will show you a couple of experiments later on, is of course not only a self-crowding, that means you have protein A, and only the same type of proteins, but there are also other types of crowding around, like for example crowding by polymer solutions, where you have the proteins which are here depicted by these black dots here, and then you have the polymeric crowders, and these polymeric crowders can be small, they can be large, and if you go to, so to say, to an entangled solution, then you can also have a network of these polymers, and inside that network these proteins are diffusing. And it's clear that the network structure, the network properties are of course affecting the diffusion, right? So the question is how that size here and how interactions between the proteins are modified by that. And there's one very famous interaction, which is the so-called depletion interaction, which is, for example, in that case, an attractive interaction. So if you think about, you have two proteins here, right? Protein A, protein B, same protein species in that case, and then you have polymers surrounding, then there's something which is called an overlap volume, and in that overlap volume here between the proteins, this is an excluded volume where no protein can enter. And that results in an attractive interaction because the osmotic pressure by these polymers here around is larger, and it's causing an attractive interaction. So same thing is depicted here, right? So you have protein A, you have protein B surrounded by these small dots here. And if these proteins are now coming closer to each other, they're producing this excluded volume here, and the result is that you have an osmotic pressure which brings these proteins together. Right? That is one of the examples of interactions which are caused in such a crowded environment. There's another funny type of interaction which is not very well known to most physicists, and that is what is called hydrodynamic interaction. So when proteins are moving in the cell, they are not moving in vacuum, they are moving in water, right? So the hydrodynamic action is basically the interaction which is mediated by the solvent. So when protein A here is moving in that direction, it is creating a fluid drag onto all the other particles, right? So this is an interaction between all of those particles mediated by the fluid, by the liquid water. And that is a complex interaction, right? It depends really on the position of all of these atoms and proteins, and it depends also on the exact arrangement of these things. So it's a many-body interaction that makes it complicated from the beginning, plus, and that makes it complicated for everybody who likes to do simulation, it is long range. It is decaying like 1 over r. Right, so no exponential decay which you can cut off with a screening length. So this is really decaying on a very long time, on a very long range interaction. Right, and the result of this hydrodynamic interaction is always a slowing down. So it's not only excluded volume; it's also this hydrodynamic interaction mediated by the solvent. These are key type of interactions which are, so to say, happening here, and. What we would like now to understand in our experiments is this process here. So we are looking into concentrated solutions. So we have a kind of cage here. So we have a protein here, and then it's surrounded by its cage of neighbors. And then there are 3 different time scales. There's a short time interaction, where a short time scale where this particle is just bouncing in that cage here, and then just a little bit exploring that cage. And the short time is, so to say, any time scale which is faster than a microsecond. So short time dynamics is usually 100 nanoseconds, 10 nanoseconds, where that particle is just exploring its cage here and is, so to say, talking to its neighbors. The short time dynamics has been very well understood. More complex, and why we are interested in, is now the cage time, where that particle is exploring its cage here, and even the long time where that particle is moving out of the cage, right? And if you plot the mean square displacement of such a protein as a function of time, then we find something like a short time dynamics, which is proportional to t. Then there's a subdiffusive motion where the particle is tracked here, and then there's a long time limit. That needs to be understood. And this cage time and the long time are really, really difficult to track by traditional biological methods. And this is where our large-scale facilities come into play now. So we are using a method which we have developed here together with FIVOS, which is called XPCS, and that is the short for X-ray photon correlation spectroscopy. So we are using X-rays and shine these X-rays onto our protein solution. And because these X-rays are coherent, they are producing, when they are falling onto that sample environment here, they are producing something which we call a speckled pattern. So you can see that this diffraction pattern is not smooth, but it's a little bit grainy. And that is because it is a completely coherent beam. So this grainy speckled pattern here is reflecting the spatial fingerprint of the proteins. And if these proteins are now moving, the speckled pattern is moving as well. And if you look onto the intensity of one of these blobs here and follow that as a function of time, then it is fluctuating. And with the fluctuation, because we are physicists, we would like to get a statistical useful information out of that. We are calculating the intensity correlation function from that. And for this typical motion in the protein systems here, the correlation function is usually having such an exponential decay here on time scales of microseconds. And from that you can, for example, then by just fitting this exponential, you can get the diffusion constant out. That is how that in principle works. When you look into the details of such a speckled pattern, this is here something which has been recorded with light scattering, but it's the same with X-rays. So you have a protein solution. You shine the light onto the protein solution, and then the speckled pattern is fluctuating, and you can exactly zoom into that speckle pattern here and then look onto the details of these fluctuations. And these fluctuations are basically just the fingerprint of the diffusion of the proteins. Right? That is what we analyze. And XPCS is basically just the analog of dynamic light scattering. Dynamic light scattering is a method which is very often used by many researchers in the laboratory, where they use a coherent optical laser beam. But of course, because that is optical, the wavelengths, and that means also the resolution is limited. And if you really want to go to the nanoscale, then you would need these X-rays. Okay, so then we go to the machines, which are really, really large. So large-scale facilities, that is what we do. And this is not a synchrotron, which is producing the X-rays, but this is a long linac here. That is the European XFEL in Schenefeld in Germany. And Sweden is part of that, right? So Sweden owns, I don't know, what is that, 5% or so? A few percent, right? So electrons are accelerated here on the DAISY side, and then there's a 3-kilometer long tunnel where they are accelerated and they are entering in a so-called undulator. And the time pulse structure of the European XFEL is like this, that every 220 nanoseconds there comes one pulse of X-rays. And then 220 nanoseconds later there comes another pulse of X-rays. And those X-rays are now all falling onto our sample. And the result is that within 1 or 2 days of beam time, we are having something like a petabyte of data, right? And so it is really, really important that these huge amounts of data are processed in a proper way. And so we, together with also with FIVOS and other groups, we were, so to say, establishing a data pipeline in such a way that already during an online experiment, you can already see these correlation functions coming out. Now, that was very, very important. And another very important experiment was this experiment which we conducted here together with Anita and Mario Reise and FIVOS group in 2022, where we demonstrated that these experiments with proteins are feasible at the European XFEL. Right, an FEL has a very high intense X-ray beam, and proteins and high intense X-rays are usually not a good match. So one has to come up with certain experimental setups and experimental protocols that ensure that these proteins are not completely destroyed by the first X-ray pulses, right? So what we do is now we shoot the X-rays here onto these capillaries with a highly attenuated beam, and then we scan the whole capillary in order to get a good signal out. And that was not clear from the beginning that this is working, but here in 2022 we could show, okay, you can basically obtain information from such protein solutions at the European XFEL without destroying your sample. That was very important. Good. This is information provided by XPCS. Now we have to go a little bit into the X-ray science. X-ray science is something which is happening in so-called reciprocal space. Right, that is what we learn in the solid state physics lecture. So all of the information which we have is basically plotted as a function of Q. Q is, so to say, the reciprocal length scale, so the real length scale is 2 pi over Q, but in X-ray science we always plot everything by Q. Right, so in such an XPCS experiment we get 3 different types of information. The first is the structure factor. This is the S of Q here, which tells you basically that when you have such a peak in the structure factor that you're forming the cage of the proteins. You get something like the diffusion constant out. That is exactly what we want to measure. And these hydrodynamic interactions, which I showed you in the beginning, these hydrodynamic interactions are, so to say, recovered by calculating by this equation here, d of q, that the diffusion constant is nothing else than the particle diffusion coefficient at an infinite solution multiplied with the hydrodynamic function, which is reflecting the hydrodynamic interactions, and then divided by the structure factor. Right, that is information we get out of these experiments. And as I was telling you, we are physicists, so we would like to understand how this d of q is, so to say, shaped by the interactions. And there's a little bit theory which I just would like to show you, that the dynamics of this coherent structure factor, which we can follow by XPCS, is basically given by this equation. So it is a differential equation here. So on the left-hand side, we have this differential equation of the structure factor, which tells us how the density fluctuations at a given Q evolve with time. And then we have the short-time dynamics, which is this exploring the cage here. So that's a short-time diffusion with no memory, and this is controlled just by the mobility, that's the diffusion constant. And then on the right-hand side, we have the so-called long-time dynamics. The long-time dynamics is really, really complicated. It's a complicated expression, which is basically dominated by a function m, which is the memory. And the memory just tells you that the particle, when it goes out of that cage, it remembers that there were other particles close by, and that it was basically bumping into those particles. So the long-time dynamics, which we would like to understand, is basically covered by this memory equation here. Right? And the large M means strong memory. That is when we have, for example, a glass or a high-density liquid. And when the memory function is small, then we have weak memory. And that means that the system forgets quickly that other particles have been around. Right? So particles move. They disturb their neighbors in that cage. The cage is pushing back later on. And that all leads to non-exponential decay, caging, aging, all that what we can investigate. So that is, so to say, what we would like to see as physicists from diffusion of proteins. And with that, I would like to show you a couple of examples. So the first example is the famous LDL. So if you take an egg, the egg yolk, then the egg yolk has two constituents. And one is large granules, which are size of 1 micron. And if you centrifuge the egg yolk here, then the liquid fraction is basically consisting out of these LDL particles. So there are 30 nanometer protein particles, phospholipids with a fat core, and these apolipoproteins on top of that. And egg yolk is basically a perfect example of a crowded environment, right? That is what we know from the kitchen, right? If you have an egg yolk, it's not water. It's a little bit viscous, right? And so this viscosity has its biological reason, and for us it is good because it is a really very crowded environment which we can test, right? That is on the left-hand side the egg yolk, and on the right-hand side you can also dream about making that as a nanocarrier, but I want to spare you this. Okay, so we take this egg yolk, LDL particles in the concentration here from the egg yolk, from the gas station, our €1 sample, and then we put that into the X-ray beam, and the first kind of information which we get is, of course, the structural information. So we investigated 3 different concentrations, 421 milligram per milliliter, that is a native egg yolk, and then we diluted it a little bit to have a comparison here, right? And that is a structure factor which we can measure for those 3 different concentrations, and you can see we have a very nice structure factor which has a value of 1.6, and that basically, this structure factor is basically reflecting the fact that we have this caging which I showed you before. Right? And of course this caging is a little bit less pronounced when we have less concentration, but it is surprisingly very nice caging which is already present in the egg yolk, LDL. And all of that really refers to the native egg which you can buy at every supermarket, right? So by fitting these curves in detail, you can also clearly find that the interactions between these spheres are not hard sphere interactions, so there's something what we call a soft sphere interaction. So the potential between the interactions of these particles is somehow decaying like 1/r to the power of 9. So that is some information you can already get out by fitting the structure factor here. And now we want to continue and want to see what is the dynamics. So we measure the dynamics. So these are the correlation functions for the different concentrations, which I showed you. And then you can go to these correlation functions and fit the correlation function, and then you can get the dynamic information out. And what is already clear here is that the dynamics becomes faster with dilution, but that is self-evident. Okay. What we now do, we don't use, in that case here, we don't use directly the correlation function, but instead we calculate the logarithm of the correlation function, and from the logarithm of the correlation function we get something like the mean square displacement of these particles. So inverting these correlation functions gives us this plot here. This is mean square displacement on the y-axis, and on the x-axis we have the time. And now we have our 3 different concentrations here, and you can clearly see that in this mean square displacement here, we have a curve similar which I showed you before. So in the early times here of 10⁻⁻⁴ microseconds to 200, 300 nanoseconds, we have the short time dynamics. Then we have a bending over of the mean square displacement. That is exactly where the particle cannot move any further. Right? It is somehow caged inside that cage. Right? It stops. It does not really stop, but it's slowed down. And then later on, it can escape its cage, and then we have the long-time diffusion here. Right? So that is how, out of our data sets, we can get all of these information directly: short-time diffusion, caging, and long-time diffusion. And for example, the cage here, you can directly read out the size of the cage, which is around 3 nanometer. So this cage in the egg yolk here, LDL particle, that is around 3 nanometers. So if you want to convey a completely boring physical message in the breakfast to your spouse at home, then tell them 3 nanometer is exactly what these LDL particles are exploring here, right? But that can be really quantified. Now we look into the detail of this long-time diffusion here and plot the long-time diffusion coefficient here as a function of Q, because we have this ability to map out the reciprocal space, and we compare that to the structure factor here on the left-hand side. Right? And that is true for the diffusion on long-time scales, what we are looking on here. And then if you look onto the diffusion here, then this collective diffusion, this D of Q, is not a constant, but it's clearly decaying, and it's hitting its minimum here, and then it is increasing again. And this is basically reflecting this very close correlation between the dynamics and the structure. That is something which is, of course, very well known since the times of Dijon. That is called the Dijon narrowing. That means whenever the structure is high, whenever we have a very high structure peak, we have a very low diffusion. Right? So this is basically stabilizing these cages. Right? And the important thing is now that this is not established here for the short time, but that we can measure that for the long time diffusion, for the long time scales. And now we can use this D of Q, and we can use the S of Q to extract the so-called hydrodynamic function. And this hydrodynamic function can be plotted for this egg yolk particle. And again, the hydrodynamic function is nothing else than the reciprocal space representation of these complex hydrodynamic interactions. And that is the hydrodynamic function which we get out. And the important thing about that here is that these are our data sets here, and this is the fit. And the fit is based basically on the exactly same Q dependence as the short time diffusion. Right? So the remarkable result which we got here is that the diffusion on long time scales shows exactly the same Q dependence. That means exactly the same spatial dependence as the diffusion on the short time. Despite the fact that these particles are breaking out of the cage and they are interacting with everybody else, we find something which we call a very simple scaling. So long-time diffusion and short-time diffusion, they show exactly the same type of Q dependence, the spatial dependence, and that was a surprise. Right? It was not clear from the beginning that it is like this, right? That this simple scaling here is existing. Right? And you can go one step further and investigate something which we call a memory function. So by analyzing these correlation functions in detail, you can get this memory function, which I showed you before, can get that out. And this memory function is plotted here also as a function of Q for the different concentrations. And what we see here is we have very strong memory effect. Memory function of 1 means strong memory effect. But what we don't see a very strong Q dependence. So this Q dependence there is a little bit of Q dependence, but not very much pronounced. So meaning this memory is more of a mean field effect. It does not have a very strong Q dependence. It has not a spatial dependence, but it's more of a mean field type of experiment. Right? Okay. You can bring that together with other results, but that's maybe not what I want to show you. I want to show you now the next example. So that was LDL, and the result is that somehow long-term dynamics is somehow mean field. It does not really show a new Q dependence. When we go for another protein, then that is the protein ferritin. Ferritin is a protein shell with an iron core. It's a very nice protein for us because it's a very strong X-ray scatterer, and it's also very stable under the beam. So going from the LDL, we now investigate these LDL, these ferritin particles, and that was a big part of Fivor's group here. Anita Girelli was a postdoc who was doing these experiments and analyzing these experiments. And the result in this ferritin case is basically very similar to the LDL. So we measure again diffusion constant, we look for the Q dependence of the diffusion constant, again concentrating on the long time, and what we see is that the same thing like in LDL, that the hydrodynamic function, that is this black line here, these are the data set, and this is the theory here. Again, we see that the long time and the short time hydrodynamic functions, hydrodynamic interactions, they are exactly the same, just scaled by a certain factor. So the Q dependence is also here exactly the same between long time and short time diffusion. Right, and this can also be plotted here. So that was a remarkable result, which was a little bit unexpected, because we were starting this experiment in order to see what is the Q dependence on the long-term diffusion. Can we, so to say, get new interactions out? Can we learn something? We learn, of course, a lot, but the surprising result was the Q dependence is the same. And why is that? That is because if you look onto our memory equation here, then the short-time behavior is clear, that has been explored before. But what our results tells us that the long-term diffusion has the same Q dependence as the short-term diffusion. And that means that this memory kernel here is, so to say, factorizing into something which has a very simple Q dependence, which we already know, multiplied with a mean field slow relaxation time. So the picture for why is the diffusion in the cells and in the liquid, in the high density liquid so slow, that can be basically brought back to something what I call a universal clock, right? So imagine the following scenario. You have this cage here, and now you're looking onto how this cage is opening. So these particles are trapped in the cage of the neighbors. In the short times, they are rattling around, and now in the long times, the cage has to open. But this hydrodynamic interaction now lead to the fact that the cage is not opening at one point here, but the cage as a whole has to rearrange. And this whole rearrangement of the cage is now, so to say, a collective motion, not a single particle escape, but a collective motion. And this sets basically the clock here. So the clock of this whole long-term diffusion is set by this cage escape time, by this cage opening time. And that leads, at the end of the day, to this factorization. That means the memory function is nothing else than A of Q. That is the Q dependence which we have seen, multiplied with that collective cage escape time, which is the universal clock. And that leads to the slowdown of the dynamics by 1 order, 2 orders of magnitude in these environments. So that is something which we really learned as a physicist, right? So that is something, it is clear that the diffusion in these very highly crowded environments is slowed down by hydrodynamic interactions. That's what we can clearly say. The question is, when does that break down? And for that, we were making one more experiment. So we were taking our ferritin sample again, but now we were doing a cryoconcentration. That means we have our capillary with a ferritin, and then we were cooling that to sub-zero temperatures. And what is happening then, you're forming ice, and the ferritin is, so to say, expelled from the ice, and it's, so to say, forming very high density ferritin samples. That is basically much better seen here on this nice microscopy images from FIVOS. So on the left-hand side, that's a microscopy image of ferritin solution at room temperature, and if you cool it down, it forms this structure here. So you see the ice crystals, and ferritin is not incorporated into the ice, but ferritin is expelled from the ice. So ferritin forms here these very high density pockets of protein concentrations. So you can clearly see that in the diffraction pattern. So you see that is a diffraction pattern from the room temperature ferritin. If you cool it down to minus 10 degrees Celsius, then you clearly form this very nice and very high structuring from this ferritin. And that is now a super highly dense protein concentration, which is otherwise difficult to produce. That is the reason why we use this cryoconcentration. And now we can also do XPCS and measure again the diffusion constant in this extremely high concentrated system. And now the diffusion time and the correlation time is not microseconds anymore, but because we have such a high concentration here, we are entering now timescales of seconds to 10 seconds. Extremely slow, right? And it's extremely slow because it's extremely highly concentrated. And now we can do exactly the same kind of analysis I showed you before. Right? So we fit these correlation functions, we get the long-time diffusion out, and then we can look whether the hydrodynamic function is what it is supposed to be. And now finally we see what we wanted to see. We wanted to see a breakdown of the hydrodynamic function and of the theory of long-time and short-time diffusion. And now if we now plot our hydrodynamic function here, these are the data points, and then we fit it with a diffusion of with a theory from the hydrodynamic functions, then now we see it does not fit anymore, right? So there's a deviation from the hydrodynamic function from the theory. And so at that very, very high concentration, we now can now finally say that the hydrodynamic function, that the scaling is breaking down, and long-time and short-time diffusion do not have the same Q dependence. So why is that now at these very high concentrations? The reason why that is relatively simple to understand. We have such a high concentration of proteins here that the water between the proteins has only a thickness of a nanometer. So these proteins are really grinding on each other, right? So there's only the hydration water between the proteins, but no bulk hydrodynamics anymore, no long flow happening, right? So at these very high densities, we have an extra slowing down by 3 to 4 orders of magnitude, just due to the fact that we are that these layers of protein water are extremely thin. We are breaking the bulk hydrodynamics, and then we have finally, so to say, a breakdown of that scaling, right? So that is something which you can do with those experiments, and maybe as a very last example, I can show you when you go away from the self-crowding, we can also look into detail about what is happening when you put ferritin into a spaghetti soup like this, dextrane 500 or sucrose, and what you can then see in these kinds of experiments, you can measure also diffusion constant in these environments, and then what we can clearly see, what we find in these experiments, so to say, is again, this is a diffusion constant as a function of Q, and that's a little bit messy here. I have to apologize for that. But this is sucrose here, so sucrose shows a clearly repulsive interaction. If you put ferritin into dextrane 100 or dextrane 500, then the diffusion constant looks now completely different than before. And what is happening here is that we have this attractive interaction happening between the proteins, and this is forming something which we call an intermediate range order. Basically means that these experiments tells us that we have an attractive interaction induced by these polymer coils here, and this attractive interaction is basically leading to the formation of such small clusters here. Right? And that is, of course, important to know, because when you have such an attractive interaction, that, of course, facilitates by biochemical reactions, because these proteins are staying on an average, they are staying longer together, pushed by the polymers together. Right? Okay, that is maybe enough of an overview about what we can do here with FIVOS. I don't want to go into these details anymore. So in order to conclude, what our experiments clearly showed is that it's not excluded volume which is reducing the diffusion of proteins in cells. The main ingredient is hydrodynamic coupling. So that is the core ingredient for protein dynamics, not on short time, but on long time dynamics. And the density, the high density, creates memory. We have clearly seen that memory function kicks in. And the hydrodynamics determines the structure of the memory function at medium concentrations. And it also clearly determines the long-time transport. And the long-time transport, which we see as long as we don't have too high density, this is acting in a mean field type of fashion. That is basically good news for simulators, because mean field means that they can just, instead of calculating the whole Q dependence of the hydrodynamic functions, they can just plug in a value which is reducing the viscosity. So the hydrodynamic coupling does not favor select any certain length scale, right? So it is a mean field thing. And the Q-independent slowdown, this may help to avoid getting trapped in glasses too early. I think that is also something which we can conclude from our experiments. If you want to see that, or if you want to connect that to a biological function, then one of the things, it's clear that the Q-independent slowdown basically leads to a slowdown of all Q, but not not necessarily to a formation of a glass. While if you go to very high concentrations, like for example cryo concentration, something which can happen in phase transition, you're a little bit screwed, because then the hydrodynamics becomes localized and highly frictious, and you're running out of this bulk hydrodynamics, and then the diffusion slows down suddenly by orders of magnitude. All right, and then of course in polymer solutions, we can clearly, with XPCS, we can clearly track the formations of what we call intermediate range order. That means these clusters forming here. And despite the fact that when you have polymers, that you are reducing the diffusion constant, and usually diffusion is also reducing the reaction rate, the intermediate range order, that means this attractive interaction is enabling more contact. So you're counterbalancing a little bit in the biochemical reactions. You're counterbalancing the slowdown by introducing clusters, right? That is basically the conclusion here. And as a quick outlook, what we want to do, we want to continue, of course, with FIVOS on that, because it is very successful. We have now formed a block allocation group with 15 groups internationally, from India, from Japan, from the US, to explore the vistas of these experiments at the European XFEL. And we want to go on the right-hand side to pharmaceutical relevant samples, so going a little bit away from the LDL, and then you can look into diffusion and molecular properties of monoclonal antibodies, for example, where we did first test experiments. And what we will also do is we will also extend that into pump probe. That means inside our trains here we can bring in a laser pump and induce temperature jumps, induce phase transitions, and then with XPCS we can map out the dynamics after these phase transitions. Okay, thank you very much for your attention, and looking forward for questions.
Thank you. Very nice overview. So now the floor is open for questions. Yes? You want to shout, or you want to like?
Yeah, I mean, you can hear?
Yes, we can hear you.
Thank you. This Q scheme, does it mean that diffusion is sort of, the means square displacement is linear in time, even in the large?
Yes, so for this, not always, but for those systems I showed you, it is. That. Here, yes, for the LDL, the long-term diffusion is linear in time. So it is Brownian motion on the short time, and it's Brownian motion on the long time.
But it is different effective diffusion.
Yes, these long time and the short time diffusion are different. They are different by a factor of 10. Factor of 10 is close to the rest, which is fun, right? So this is LDL, this is your egg yolk from home. And it has been designed by Mother Nature in such a way that you store as many as LDL as possible, but you're still in a liquid phase. Right? So LDL still needs to be transported to the envelope, and still needs to be able to flow around. And somehow the maximum storage capacity is when short-term and long-term diffusion are around a factor of 10. If it would be a factor of 15, it would already be frozen.
So this is for the AU and the ferritin, both of them?
Both of them are very similar in that respect, yes.
So this memory effect that is caused, I mean...
The memory effect is caused by this caging here. So,
yes, but I mean, how does it depend on the density?
generally speaking, it is the higher the density, the more memory effects you have. Right? So the memory effects are basically coming into play from the caging. Right? So you have the cage, and the stiffer and the smaller the cage, the more memory effects you have. So if you have a completely diluted system where protein is here and the next protein is over in Uppsala, there's no memory. Right? But when things are coming very, very close to each other and these cages are becoming very, very stiff, memory becomes very strong. The memory becomes very strong. It's basically showing up in the banding of this curve here, the difference between short-term and long-term.
But you also said it was due to hydrodynamic interaction. So the hydrodynamic is somehow affected also by the density.
Absolutely. Yeah. Unfortunately, yes. So this comes together. So the more density, the more hydrodynamics you have.
Okay.
That comes on board. But what is important is that the slowdown I showed you cannot be explained just by Cajun, by structure, but you need the hydrodynamics. Otherwise, without hydrodynamics, the slowdown would not be a factor of 100, but only a factor of 2, 3, 5.
Thank you. Are there more questions? Yes?
So you mentioned that you can start a phase transition. Can you say a few words about what kind of transitions and what?
Yeah, we are very much interested in liquid-liquid phase separation. Right? So liquid-liquid phase separation when you are basically coming out of the homogeneous phase and then you're switching on attractive interaction between proteins, then you have a liquid phase separation. That means you have the system separates into high-density parts and into low-density parts. Right? And that is the paradigm which is of large interest for the biological community. The physicists have been knowing that for 100 years, new applications, stuff like that. But in the biological community, it has been discovered 20, 30 years ago that this is happening really in cells. I think that you can form high-density protein compartments in the cells of the body. And understanding how that is happening is still open from the physics point. And you want to understand how that is initiating this phase transition, what are the density modes which are happening, and you would also like to understand what is, so to say, the end phase of these liquid condensates. Right? Are they glassy? Are they liquid-like? This is one of the questions we would like to answer with these phase transitions.
All right, thanks. Yes, more questions?
How long memory did you observe in your measurements?
You can see these typical time scales which we have here, right? That is answering the question. So we can measure until 100 microseconds right now. That is basically given by the machine itself. So we are sitting there at the European ZR, and the European ZR is giving us 300 pulses. And then it cools off. And then the next pulses are coming 100 milliseconds later. And we are in a very different time regime. So that is the time scale which we can cover here, from 220 nanoseconds until roughly 100 microseconds, and then we are phasing out. So if you have a system which is much slower, then you would have to go to a synchrotron. That is what we did with that cryoconcentrated ferritin. The cryoconcentrated ferritin has been measured at a synchrotron where we have much longer time scales. And then the memory is happening also on 10 seconds. It is one of these paradigms, right? That dynamics, that means viscosity versus volume fraction, is of course exploding. When you come close to the glass, then viscosity is exploding without the structure factor being exploding. So this is why you are ending up very quickly when you concentrate that even more. You are ending up very quickly in times which are seconds.
Thanks. One more question, yes?
Thank you for the talk. So for this particle in the cage model,
Yeah.
how would it be different if there are heterogeneous species, let's say different proteins of different sizes, and is it something of a relevance to look at?
Yeah, this is of relevance, and we did the experiments, but they have not been finally analyzed. So of course there is to be expected a size dependence of these cases, right? So large particles versus small particles, we will expect a difference. That depends also on the relative volume fraction of large particles versus small particles. Right? So we are working on that. I don't have the final answer now, but we already see that there are effects like this happen.
Thank you very much. Is there any last questions? We had a very nice discussion. Let's thank Christian again.